Okay, thank you, everyone. Welcome to our talk in the Maths and the Life Sciences / Northwest Seminar Series in Mathematical Biology and Data Science. It's a pleasure this week to be joined by Professor Ruth Baker from the University of Oxford. Ruth is a professor of applied mathematics, works in areas around mathematical biology, particularly applied to developmental cell biology, which we'll talk about today. I think in terms of maths, you cover a broad range, right, of techniques and collaborate with people from across disciplines, but I'd say a lot of the applications are in this developmental biology area. So it's great to see so many people in person and also online. This talk is being recorded, and so that will be made available later as well if people, anyone listening to me now obviously could make it, but people want to watch it again, you can catch it online. So without further ado, hand over to Ruth.
Thank you. Thanks, Carl, for the invitation, and to everyone for joining. So you can see the title. There's going to be two parts to the talk, really, or two aspects to it. Number one is to talk about what it means to have an identifiable model, why we might want identifiable models, and if we have identifiable models, what can we learn about how the cell cycle is regulated. So everything today is going to be quite simple models applied to quite simple data, hopefully to illustrate some of those points. And I'm also going to try and highlight that we where we got it wrong along the way and how we didn't really realize that we'd got it wrong until we took existing models and applied them to new data sets. In particular, the kind of key sort of data that I'm going to use today or to think about is data like this that's on the slide, and this is data that's collected where the cells have what's called a feature reporter. So they're engineered to have this feature reporter in them. And what does that really mean? Effectively, in the kind of early part of the cell cycle, G1, then the cells express a red fluorescent protein. And then in the sort of SG2M, I'm trying to indicate with the mouse, but I realize you can't see that. In SG2M, then they express a green fluorescent protein. So if you look at the data, there's often a little bit of short periods between G1 and S where they can look a bit yellow, where there's both around. And then immediately after mitosis, they often just appear dark, so there's no expression. And you can see in this, I've got a bit of a figure from the original paper, just how well that allows you to visualize across tissues the parts of the cell cycle that cells are in, and you can begin to understand how synchronized the cell cycle is, for example. And so our idea was to understand or to think about how to use data that's collected with cells engineered with this reporter to try and understand how cells might progress through the cell cycle, and how that might be how that progression might be regulated by kind of local crowding. And so they are going to be really simple models. I think I already said this to Tash. There's going to be no mechanics or anything in there, so we're just really thinking about cell numbers or cell density most of the time. Okay, so I think I've just said precisely that. So that's the sort of the question for today. Okay, so I am going to use PD models, but they're going to be very, very simple models, just looking at essentially how the density of red cells, if you like, and green cells evolves over time. And so there's going to be reaction diffusion equations, and I'm going to be interested in doing parameter inference, and oftentimes, not exclusively, I guess I'm interested in a Bayesian framework. And the reason for that is that effectively what I can do is I can gain estimates of the parameters, but also learn something about how tightly I can pin them down, right? So I can quantify the uncertainty in those parameter estimates. And so I'm targeting the posterior, which is the probability of parameters theta in my model given data D, and I write that in terms of the product of the likelihood and the prior. And just as a bit of a note, so sometimes we think about the likelihood as essentially the likelihood of observing the data given the model parameters. And so that's sort of thinking about a distribution in D. And sometimes I think about it as sort of saying, given a fixed data set, what's the likelihood almost like it's a function of theta, right? So you can sort of think about that exchangeably, and I'm going to do that just a little bit here. And I'll try and point that out or be clear what I'm talking about. And so if I have a PDE model describing the evolution of cell density over time, then essentially if I have an error model, so that might be sort of noise relating to the kind of the process itself, or that might be the noise that I have in my measurements. If I have this error model, then I can access the likelihood essentially because I can generate the numerical solution of the PDE model, and then I can convolve that effectively with the noise model. So I'm going to do that today, and that allows me to write down the likelihood. And I'm going to use additive Gaussian noise because it's really, really simple, because that effectively just entails a log likelihood that looks like the sum of the squared differences between the data and the model. But you can repeat any of this with a different error model as you want. Okay, so from a from a kind of theoretical perspective, the sorts of questions I'm interested in, well, I'm sort of interested in asking, given a particular data set, almost like what's the relevant level of complexity of the model for that data set? So is my model at the right level from the data? And so questions I might be asking are the parameters identifiable? So in principle, given access to lots and lots of very, very good data, could I estimate the model parameters? I'd also care about, given real world data, can I estimate those model parameters? And can I, again, quantify the uncertainty in those estimates? So how sure am I about what I'm estimating? And the reason I really want to care about that is because I care about how that uncertainty in my parameter estimates then propagates through into uncertainty in the predictions I make with my model. So if I use my model to make some predictions that I want to test experimentally, I'd like to be sure of the range of possible outcomes that I might be expecting. A big one for today is going to be how, if I change the experimental setup, and actually for me, it's going to be only really just slightly, there's no big changes here. How does that really impact my ability to measure parameters or estimate parameters in a model? And then after we thought about all that, can we learn something about the cell cycle and regulation? So that's the kind of idea. So here's kind of what my data looks like. Sorry, it's not particularly easy to see. Just see that. I like a little bit. We probably don't need to see much. Yeah, much better on the Zoom than on screen. Okay, I've basically got data from 2 assays that I hope you can see. They're different cells. They are basically cells. So on the top one, I've got what's called a barrier assay, where you grow cells, seed cells inside sort of like a barrier or a cookie cutter, and then at time 0, you remove that barrier and you just look at the cells as they proliferate and they migrate outwards. And what you can hopefully see here is that you start with something that's approximately fairly homogeneous in terms of red and green, so distribution of the cell cycle-ish. And over time, what you see is that predominantly you get a green rim and a red middle. It's not quite that clear cut, but that's sort of what we're seeing. So I've data from our collaborators in Princeton, it's published in eLife. And then this is a study from collaborators published in Biophysical Journal, where they did a very similar experiment with this feature reporter. So the top is MDCK cells, I think. The bottom is, I think it's 2-3 more breast cancer cells. But the point being that this is here, the experimental setup is slightly different. So it's a scratch assay. So you grow the cells in a dish to whatever kind of sort of level of density you want. And then at time 0, you scratch away a portion of the cells. So it's called a scratch assay. And so you see this area here that's initially devoid of cells, and then over time they migrate into the space and they proliferate to fill the gap. Sometimes called a wound healing experiment. I mean, yeah, it does look much better on the screen. I'm sorry. Okay, so these are the kinds of data that we are going to use. And in particular, we're going to make it simpler than all of that. So instead of having to deal with a 2D problem, in particular for the scratch assay, we're just going to average everything in the Y direction. So we get a density profile in X, which is across the wound over time. And we are going to oftentimes count the density of reds, or count the numbers of reds and green cells separately. So we get these two profiles. So that's the kind of data we have. Okay. So the first question you might ask is, if I had a model, what parameters could I sort of hope to be able to estimate using these kinds of data? And so the first thing I want to talk about is the concept of structural identifiability. And I think I wrote it on this next slide, but structural identifiability is kind of like, it really only thinks about the model and not the data. And it just says, if I had access to perfect data, essentially, so here, all space X at all time T, could I estimate the parameters of my model? And really the question is, do distinct parameter values or parameter sets in my model always give rise to distinct output distributions or distinct distributions of observations? And then if that is the case, then it means that essentially, if I have this perfect data, I could estimate the model parameters. So clearly it doesn't take into account the limitations of the data. And you can see from that previous slide that this data is generally not noise free. We can't collect it. We collect about every, I think 6 or so, not quite, hours in this assay. So it's definitely not error free. It's definitely noisy. It's incomplete. So we don't have that case. But I think for me, the reason why we want to understand the concept of structural identifiability or the property of the model is that if we don't have structural identifiability, we will never be able to estimate the parameters uniquely of our model, no matter how much data we have. So it's a useful first place to start. And there's lots of work developing methods for ODE models, but there hasn't really been a huge amount for PDE models. So one of the first parts of the story I want to tell you about is some work we did recently trying to extend the concepts that have been used to think about ODE models into the land of PDE models. And it really isn't, it really isn't conceptually more different. Okay, so how does it go? There are different ways to do this for ODEs, but one way of doing this or tackling this question of structural identifiability for ODE models is to think about what we call the input-output relationship. So this is a really simple model. I don't even want to talk about what people think about it representing. It's not very relevant. I think I've got variables Q1 and Q2, parameters P1, P2, P3, and P4. And then U is a control input. So I can vary U as make U a function of time, and I can choose what that is. And then what I actually observe of my system is Y, which is Q1 over VP. So VP might be some volume or something. So if I want to think about structural identifiability via this input-output relationship, then what I need to do is rewrite my ODE model in terms of the observed variable. So effectively, there's some differential algebra where I differentiate and I substitute, and eventually, after a while, I get down to an ODE that basically describes how my output evolves in time. So it's a higher order equation, and it contains essentially various combinations of the parameters as coefficients. And what you need to do, and it happens, just so it happens here, is you need to always make sure that one of the terms has got a coefficient of 1. Otherwise you can rescale your sort of coefficients. But the idea is that once you've done that and you've got this input-output relation, then if you have perfect data, you can estimate each one of the coefficients in front of these terms. Okay, and so that gives you the set of combinations of parameters that you can estimate. So here I can get VP times P1 plus P3. I can get this kind of slightly more complicated combination of parameters here from Y, and then I can get P3. So what does this tell me? If I put in a time varying U, then I can estimate P3, I can estimate VP, and if I can estimate those two, I can also get P1. Then I've got this product, but I still can't get those two. So out of this model, if I just measure Y, then the combinations of parameters that I could ever hope to learn would just be P1, P3, and the products of P2 and P4. Okay. So that's the kind of idea behind thinking about input-output relationships and how they enable you to determine what parameters you might be able to measure. And one of the things you might want to do from here is then to reparameterize this model. So just consider the product of P2 and P4 as something to estimate. So how do you take this approach and how do you use it for PDs? Well, in essence, it's not really any different, but the algebra is just harder. So thinking about this data that we had before, we're just going to write down an incredibly simple model and see what we might be able to learn. So we've got red cells and green cells, so density is a function of space and time. And they diffuse, coefficients D1 and D2. And then in my model, basically K1 is a parameter that represents cells progressing through the cell cycle from G1 to SG2M. So they're transiting from being red to green. And then K2 is the rate at which cells are progressing from being green to red, right? And the fact that cells divide as they go from green to red gives you the factor of 2 in the first model. So this is like the most simple model that you could write down of this system. And one of the questions you might ask is actually, do I need to measure the density of red cells and green cells separately? Or could I estimate all of the parameters of this model just by looking at the total cell density? Because that wouldn't require me to have this reporter in the cell line. Okay, so the question is, can we reduce that model to an input-output equation in this variable n? Because again, then these coefficients are going to determine these identifiable parameter combinations. So it's messier because you have partial derivatives. So we just use an ij to refer to the ith derivative in space and the jth derivative in time. But you can do it, and this is what it looks like. And it's monic in the sense that one of the coefficients is 1, and then you can just read off your identifiable set of identifiable coefficients. So if I have perfect data n, I would be able to measure these coefficients. And if you just look at it quickly, you can see that if you can get the product and the sum of the rate parameters, you can get each one of them individually. And if you can get the product and the sum of the diffusion coefficients, then you can get each one of those individually. So in theory, well, this model is structurally identifiable, which means that in theory, I could estimate all the model parameters just by measuring the total cell density. So that's a sort of useful thing to know. So it's a very simple model, it's linear, but I can still deduce that. So in this paper in Proceedings A, we could show that more generally, if you have a coupled system of reaction, diffusion, advection equations that are linear in the observed variables, then you can always establish that input-output equation. So you can always reduce it to it in, or to see how to put in your own equations and to generate those input-output equations. And there's definitely work on the archive as well by Heather Harrington, amongst other people who have a more sophisticated differential algebra approach that can do this for more complicated equations than we can. So I think it holds more generally and for reaction diffusion equations. There you go. Okay. So we know something about how to establish whether, in principle, we can estimate our model parameters from the data. And we can play that game. And in fact, for all the models I'm going to talk about the rest of today, we're going to measure both the density of red cells and the density of green cells. So structural identifiability is always a property of the model. So we're going to write down. And so really, we're interested in then, actually, if I have real world data, how well can I do? Okay, so if I have real world data, how well can I do is what we mean by practical identifiability. And it's much, I think, less well defined than structural identifiability, right? So my slightly ad hoc definition is going to be to say, can I provide reasonably precise estimates of my parameters using finite non-ideal, so the data that I actually have available to me. And I could put a bit more of a firm hat on that, but that's generally what I mean. How much do I learn maybe as a result of having done an experiment over and above what I already knew? So let's go back to this original data set and ask some questions of it. So I said I was going to learn, first of all, about regulation of the cell cycle, but actually I'm going to maybe do something slightly different with this data set first and ask whether this concept of the go or grow hypothesis is a relevant question for this data set. So the go or grow hypothesis is something that's often postulated in the context of cancer cell invasion, and basically sort of postulates that cells are either motile and not proliferating, or proliferating and fairly static, right? So that's the kind of idea behind this go or grow hypothesis. So we asked the question, given this data set where I'm using literally this average data, so just data in X averaged in Y, can I detect, or is it sensible to write down a model where my two cell types, if you like, or cells in G1 and cells in SG2M have different diffusion coefficients? Can I sensibly do this for this data set? All right, so what's the model that I write down? So the first model I write down, they're super simple, same structure as before really, says that my red and my green cells potentially have a different diffusion coefficient when they're in G1 versus SG2M. So when they're expressing red versus green versus protein. I slightly interchangeably, and I apologize for this, use K1 and K2 and KR and KG for the rates of progression through the cell cycle. But the question is really, can I motivate having this model for that data where the cells have different speeds, or should I actually stick to a simpler model for this data set where the cells essentially move at the same rate or have the same diffusion coefficients, no matter what phase of the cell cycle they're in? So really, the question is, are both of those models identifiable practically, given these data? And should I use practical identifiability as a means to pick one of those models over the other one of those models? And again, I'm going to think about this just in terms of these different parameters. Okay, and again, like I said before, I'm just using normally distributed error additive. Okay, so what's the first thing I can do to think about practical identifiability analysis? So I guess what I can do is just sample from this posterior distribution using something like MTMC, and then non-identifiability, so the sort of a lack of ability to really be able to estimate model parameters, usually manifests itself in terms of poorly converging chains. They bounce around in parameter space all the time, and I get broad posterior distributions. So let's have a look at what these posteriors look like for these parameters and that data. So you could do a really, really good job about pinning down the parameters that relate to rate of progression through the cell cycle. And hopefully you can see this, the lines are a bit thin, but you get really quite precise estimates, and you can put down essentially a confidence. But when it comes to these two different diffusion coefficients, those changes just bounce around all over the place. They really don't settle very well. And you get these very, very broad posteriors that go right from kind of 0 to past 1,000 in whatever units we use. I'd have to check. So I was going to say, so really we can't get precise estimates, right, for these diffusion coefficients. But nonetheless, if I do a posterior predictive check, if I sample parameter values from the posterior and simulate the model, I can see that the model well predicts the data. So I would say that this model is not practically identifiable, or these parameters aren't, and I'd maybe have some worry about using it in unseen settings, because if I pick a particular value for each of those parameters, dr and dg, I'd not have so much confidence about my predictions going forwards, or using them in context. On the other hand, if I look at that second model where I basically just said that there's just one diffusion coefficient that's common to both of the populations, you can see that I retain this really good identifiability of both the rate parameters. I can estimate them really quite confidently. But I can also now get a really confident estimate of the diffusion coefficient as well. And despite the fact that the model is kind of reduced complexity, got one fewer parameter, the posterior predictive check shows you that it's still doing a relatively good job at predicting the dynamics and the data. So I'm still happy that it's a sensible model. So this model is practically identifiable, and I would probably prefer to use that one over the other one. Or maybe I would think about how to design an experiment to better test or validate the use of model 1. Okay, so we can draw that conclusion, but actually using MTMC is not necessarily a particularly efficient way to do things. And I think another way that you can get at the same conclusion, if you like, is to use an approach called the profile likelihood. So what the idea is that you consider the normalized likelihood, and then you take your parameters and you partition them into one or maybe two parameters that you're interested in. So it might be the diffusion coefficient. So I keep that as my interest parameter. And I call all the rest of them nuisance parameters. So KR and KG would be nuisance parameters. And the idea is that what I do is I run over a whole grid of values of that interest parameter, and I optimize this normalized likelihood over all the nuisance parameters. So it's sort of like looking at the likelihood surface. If I had a likelihood surface, looking at a profile, like sort of as a set of a mountain ridge or something. And then non-identifiability, and I'll show you figures now, would manifest in there being a flat likelihood parameter. So the likelihood profile, the likelihood surface is very flat, local to the maybe the MLE. Okay, and these are what these profile likelihoods look like. So what I've done in this figure is scanned over lots of different, taken a fine grid of values of this parameter KR, and for each value of the parameter KR, I've optimized the fit effectively of the, I've calculated the normalized likelihood optimized over the other parameters, so optimized over KG and the diffusion coefficient. And what you can see is that in looking at it in this direction, the likelihood is nicely peaked around a particular value of K. And again, we can use the concept of sort of confidence intervals to put down this is like a 95% confidence set, this parameter. And if I look at the difference between model 1 and model 2, right, all the same results, right, are still there. We can estimate both of these sort of rate parameters really confidently, right? The profile likelihoods are really nicely peaked. We've got finite confidence intervals set. But if I compare the profile likelihoods for the diffusion coefficients, I can see that in model 2 again, where I just have one diffusion coefficient, this curve is really nicely peaked, have a confident estimate. Whereas in model 1, those profiles are really broad. And in fact, I don't have a my confidence intervals aren't contained within the range of values I think are relevant. So this would, and I think I've got a box around, this is where I would really begin to distinguish identifiability. And for me, at least, the reason that I would prefer profile likelihood analysis over MTMC is that this is a relatively efficient thing to do, at least when your model doesn't have too many parameters, because it's just repeated kind of numerical optimization, whereas the other requires maybe some MTMC that can take quite a long time. So I think you get the same kind of information out of both, but generally speaking, I think this is a very informative thing to do. Okay. So that's what I just said, it costs less. Although I have to say that if you get up to models where you've got, tens of parameters, then this also becomes something that's non-trivial to do. Yeah.
You're saying you need to be able to kind of normalize speed likelihood properly.
You don't have to, but because you'll always see the same peak, right? You're just making a rescale. But I guess it's useful if you want to put like confidence, look at confidence intervals, because then you know that you draw a line at a particular level and that defines your confidence bound. Yeah, so you can do it whichever way around and then just unpick it afterwards. Okay, so the take-home message from this bit is that both of the models in principle are structurally identifiable. So if I had enough data, I'd be able to estimate all these parameters. But actually, given the kind of data that I actually have in practice, I should probably prefer the simpler model, model 2, because I identify the parameters. Yes, Oliver.
To be clear, in model, the later PD evolution, you have so 1 minus R minus J suggests that they are dimensionless.
Yes.
What about the parameters themselves?
Yeah, so this is a really good point, and it's going to, for want of a better expression, come back to bite me later in the talk, right? And in fact, this is why a lot of this we didn't notice the first time. So what we have implicitly done here, or the step up from the previous model that I showed you, was just to say that we think that in a kind of Fisher KPP spirit, we think there's like a sort of logistic growth type behavior here, where the cell will only actually divide if there's space for it to divide. So this 1 minus r minus g just captures the fact that the cells will only divide if there's space, and that their rate of progressing from or through mitosis is basically inversely proportional to the space available. It also assumes under the hood, and this is really good you asked this because I sort of need to point it out. It assumes under the hood that actually what we've done with the data is to already kind of semi-non-dimensionalize this to rescale it with what is confluence, essentially. And so this just means that this one here is the density when cells are at confluence. And in fact, what we did was we just looked elsewhere in the assay or at other estimates and other assays we had, and we used that to rescale, and that got rid of a parameter. And it's quite an important point. Yeah. Yes. So several years later, it came back to bite us, and I will show you precisely why in just a second. That was a really good question. Okay, where am I at to? Yes, so I think it's important that we consider the limitations of the data in building the model. And I think carrying out this type of practical identifiability analysis and structural identifiability analysis, whilst we're sort of building the model, is quite important. And you can do that with synthetic data, right, that mimics experimental data as well. Okay. So yeah, this is precisely the point. So let's take a closer look and ask the question, is that model consistent with other data? So these data from a lab at Daniel Cohen's lab in Princeton, who we collaborate with, and it was published in eLife in 2020. This is work by Matt Heinrich. And the very short story about how this whole project came about is that one day I said to one of my PhD students, oh, why don't you just take the model we had in that previous paper and go and apply it to data and learn how, you know, cut your teeth and learn how to estimate model parameters in a slightly different data set. And he came back a few days later, was like, yeah, it doesn't work, right? The model doesn't fit the data at all. And I don't know if you can kind of see why, although it's harder to see it on the screen here than it is on my laptop or on Zoom, I think. But the problem is that we're getting a load of green cells around here, around the edge of the colony as they spread. And we've got predominantly red cells in the middle. So cells are getting kind of stuck in the first part of the cell cycle. Whereas our previous model predicted that cells could kind of only get stuck in a density dependent way in the second part of the cell cycle. But we're seeing the actual opposite thing to here. And so, it's completely right? This previous model we had just didn't explain this data set. And we didn't really notice the first time around. Okay, so here's the data set. One thing I love about this data set, and we really need to do more, is the fact that we can pick out every cell in the assay, and we can label it as being in G1, SG2M, or in grey is the post-mitotic cells. So one of these days I want to do something with this fully cell scale resolved data, but what we did was we just looked at it in terms of a density profile out from the center of the colony. And so this is good.
Sorry, I was just wondering, do cells that sort of go into G0, are they grey or do they still express red?
So my understanding is that that isn't happening, but I could be wrong. So this is just grey was post-mitotic, but don't quote me on this. So yeah, so it's something we've been thinking about a little bit more recently, because we've been thinking more about kind of the concept of age structure in this context. And we can definitely talk more about that later, but for here we're assuming not. And in fact, we're going to lump the grey cells, the post-mitotic ones, in with the G1 cells, so that we still only consider 2 populations. So we did look at 3 populations, and it just gets more complicated and we don't really learn much. But we've got distance from the center of the assay and density measurements. Okay, so then what the student Carlos did was said, okay, if we want to have a chance of actually getting our model to replicate these data or predict these data, then we need to have this sort of density dependent feedback on both of the transition terms, right? So you have a basal rate of moving from G1 to SG2M, which is K1, and a basal rate K2 of going from SG2M back into K1, back into G1 with a cell division. And those basal rates can effectively be perturbed by a crowding function. And there's two crowding functions, F1 and F2, and they just depend on the total cell density. And to make things as simple as possible, we just say that there's two additional, so they're linear functions of the density. So effectively, the rate of going from G1 to SG2M becomes 0 for capital K1, and the other parameter, capital K2, represents the process of mitosis. So mitosis stops basically when the density is K2. So now we have the diffusion coefficient, we have these two rate parameters, lowercase k1 and k2, and these two capital K1 and K2, which are these feedback functions. And we wanted to know if we could estimate them all. And essentially, one thing that you can sort of predict, and I'll come back more to this, is that the sorts of patterns that you'll see in the tissue or in the colony will vary quite a lot depending on the relative sizes of K1 and K2. Okay, so when Collis did this, he fitted the model to the data. So you can see the model data fits to the top. It's not so great, like slightly behind the edge, but we do really well, I think, at the leading edge of predicting the sort of dynamics of the invasion process. And we do get really quite confident estimates of all of the model parameters, so we were able to do a reasonable job here. But the real take-home message, I think, actually comes from looking at these capital K1 and capital K2. So the fact that K1 is smaller than K2 means that if you like, the crowding based or the mechanical checkpoint from going from G1 into SG2M is stronger, right? So cells are kind of stopped from making that decision at a lower cell density than cells are stopped from undergoing mitosis and dividing. And in some senses, maybe that makes sense, right? Because you don't want to go through all the hassle of putting the machinery together to be able for a cell to be able to divide if that's not going to be possible. So the prediction is that that's the stronger checkpoint. And one of the useful things that you can do with this then is you can make predictions of how long each of the phases are. Yeah, so we did put G0 in there. There you go. I'm not smiling pressing that. How long each of those phases is as a function of cell density, and then we can also predict sort of how the cell's total cell cycle length depends on the local cell density, which was good. Oh yeah, do you want to? Can you see it? Do you want to read it out?
It's just about the is asking if any of this is related to the individual cell size, like because the densities of the cells per millimeter squared, is there an effect of cell size?
The short answer, we didn't look at it here, but in a different study, we had an individual based model and we let the cell radius increase over time. So we parameterized that just from looking at experiments. So we did, so in the experiments, we knocked out cell division by applying like mitomycin C, but the cells still round up as if they're going to divide, so they still change size. And we looked at comparing the fit of the models where the cells were constant size or the cells were changing in size, or the cells couldn't see each other. In fact, we looked at comparing those fits to data, and it turns out, yeah, I mean, it makes a difference, but it's probably not like an order one, as it were, effect. But yeah, no, that's a relevant question, and we could certainly go back and look at that. Were there other questions?
No, I think that's
okay, cool. I'll have a look at them after. So one of the things that had sort of been bothering kind of as he did the study was that this idea of there being two checkpoints was a kind of theoretical thing, and we weren't really aware of any literature that backed it up. But after a bit of research, then we already knew about the Strickland paper, but we found this paper that was recently published in-cell reports that basically provided justification for the second checkpoint. So it was a little bit of a relief to be able in the paper, right? Oh, actually, this is consistent with what people are seeing experimentally. So that was nice. Okay. So what else can you do? I mean, these models are sufficiently simple that one of the nice things is that you can do a standard traveling wave analysis to look at how the wave speed depends on the rate parameters. And actually, one of the things that in essence, we should predict, right, is that when we linearize at the wavefront, we're doing that looking in the low density regime. That means that those crowding parameters are not relevant. They drop out of the analysis. So the wave speed is only predicted to depend on those rate parameters lowercase k1 and k2. But if you do numerical simulation, you can really show how the sort of structure inside your wave invading wave really heavily depends on those sort of crowding parameters. So they are important kind of back from the wavefront, and then think in terms of thinking about what's going on inside. Yes.
Sorry, if you compare these solutions you get for these wave speeds with the observed behavior with the speeds of the parameters.
Yeah, so it's pretty good. So if I go back to these kinds of models, so then definitely solving the full model, we get a good agreement. Arguably, we get the best agreement at the wavefront, and then there's some slightly spurious behavior, and there always is, I think, right towards the center of the colony. And no matter what study we work with, and no matter whether we're looking at future or not, there's always something that's happens at the middle. But I think the front is pretty good, and that and these estimates are good. Are a good prediction of what you see when you solve the PD, as long as your initial conditions are sensible. Yeah, yeah. I mean, I guess as long as you as long as you can measure often enough, yes, you'll get some kind of measurement. But you don't certainly fit the wavefront profiles very well. If that makes sense. So it's not too bad. I mean, go on, yeah. Sometimes we just that's just how often images are taken. We've got other experiments where we've got data every 5 to 6 minutes. But so there's a real trade-off, right? It's two things really, right? It's great to have that kind of data, and in that kind of context, we have thought more about how to learn the model directly from the data, or to how to parameterize models on a much bigger scale. One of the real issues here is if you think about what happens to your likelihood as you add more data points in, is that it gets very, very small, and it's much harder to work with. And you don't, there's definitely very quickly a point past which you don't learn much more by adding in more data points, right? You don't get more accurate or more certain predictions, but it takes much longer to run, and it becomes computationally infeasible. So I think there's a bit of a balance between how much data is useful and how much more do you learn, if that makes sense. But certainly we've got data from like a rubber stopper assay. So instead of making a scratch, you put like a rubber bung in the middle of the dish, and you take it out, and then you watch as the cells fill the gap. And there we can track every cell, like 10,000 cell tracks, and we're doing something slightly different with that, but depends on the scenario. I think this works not too bad here. I was going to say something. It might come back to me. Oh no, it will in a minute. I think. Yeah. So having said all that, one of the interesting things is to go back and to think about other data sets in the literature, and think about how valid the model is for those other data sets, and what you have to be careful about. So in that Stryken paper that I flashed up before, what they actually did was they grew their cells to a very, very, very high density before they, I think it's actually a scratch assay they made the scratch. And so all of the cells pretty much at the point at which the scratch is made are like stuck in G1, or even G0, I guess. And so when you make the scratch, it takes an awful long time before you start to see a strip of cells at the front, they're actually green. So they're actually sort of cycling, and then they begin to proliferate. And so I think in this context, the initial conditions are very, very important in the predictions and to understanding how long it is before you get these kinds of patterns. And also here, I think, so this is just taking the parameters of our model and applying it to initial condition that was like in the striking paper. So we didn't re-parameterize the model. But I think it's quite difficult, given access to data like this, to nail down both those capital K parameters, because I think that the signature's just not quite there, trying to say. So we could predict, it was the same cell type, it made sense to predict. And we could predict reasonably well, at least as a qualitative level, but I think we would have struggled to get all of the parameters out of that data. Going back to Oliver's question, and why we didn't pick things up before. So when we first did this study, so it was the one published in Interface, yeah, so what we did was we basically non-dimensionalized our cell densities with what we thought a confluent density was. So cells in the model could go from density 0 to density 1. And the kind of knock-on effect of doing that is you don't have to estimate that carrying capacity parameter. Okay. And so when we wrote down our model, the model we actually used in that paper was one where in here we had a sort of 1 minus R minus G term. And we never really noticed that we didn't need it, right? And we didn't need it because we never really noticed, because effectively 1 minus R minus G was approximately 1. And so that the loss of that term, or having that term in there, really makes very little effect. And so it actually turns out for this data set, you can just as well fit that first model that I showed to the data where there's no kind of regulation of the cell cycle by local density, and it fits the data just as well. And the reason it does that is that actually these were experiments, and we never thought about it, that were conducted when the cell population was initially at really, really low density. And had we, I think, not non-dimensionalized the cell density, we would have picked that up. We would have picked that up much, much earlier. So there's like a subtle difference, and it actually just, there was quite a big knock-on effect. Yeah, and then there we go. And the other thing I wanted to say is that what's always a problem with these assays is that when you make a scratch, it takes on the order of like 12 hours, I think, for the cells, sometimes longer, to really start moving and proliferating and acting in a quote unquote sort of normal way again. So I think we're always fitting slightly the wrong model to these data. And I think that somehow impacts some of the results, but that's maybe a story for another day. So take home messages. I think the use of quantitative data really allows us to then quantify these ideas of crowding or density dependent regulating of the cell cycle. And there, I think our study shows that there are at least 2 checkpoints, which are, I mean, mechanically is maybe too strong a term here, that are regulated according to local cell density. And I think you could repeat this kind of study if you had something like a 4-color Fucci equivalent, as long as you had sufficient data. So you might be able to break this down a bit better, but it would depend on the identifiability of the model as to whether you could get anything meaningful out of it. We tried with 3 and we didn't learn any more, so I'm not sure. But the other thing I wanted to highlight, that experimental design matters, and I think we should think about the limitations of data in building the model, and we should really think about, when we compare model with data, what we're actually comparing, because there were some subtleties hidden in that it took us a very long time to notice. And then that you can also learn new things by exploring existing models with new data sets and vice versa. And then just to say thank you to people who are really involved in this work. So a lot of the study was done by Carlos Falco, as a student in the group, so that's finished. Maria and Alex did lots of the structural identifiability work. Jose co-supervises Carlos, and there's lots of other people in and amongst that who've contributed. I just wanted to say that there's a sort of mini tutorial paper that, fingers crossed, is going to be to go in the SciAm review, come out in SciAm review, or be accepted in SciAm review first would be a good one. But it's on the archive, and if you want to look at it, then it's there. But we've got all the codes in Julia to do all this practical identifiability stuff, if you like, and there will be Python code as well, because that's one of the reviewers' comments. So please do have a look at it, and I'm happy to take. Thank you for listening. I'm happy to take any more questions.